Hello students, welcome to the EPG Pathshala. I am Dr. Rajeshwari Sinha, working as Program Officer at the Center for Science and Environment. Today, we are going to learn about DNA cloning technique, an immensely important technique used in the field of molecular biology. DNA cloning is a technique to reproduce large number of fragments of DNA of our interest. This lecture will have broadly 3 sections. First, we will have a brief introduction to the process of DNA cloning. In the next section, we will look at the various steps of DNA cloning and then see the applications of DNA cloning. There may be many different meanings for the word cloning. Basically, cloning means cutting a piece of DNA from one organism and inserting it into a cloning vector where it can be replicated by another host organism. Sometimes this process is called subcloning because only a part of the organism's DNA is being cloned. Clones are genetically identical organisms or molecules derived from a common ancestor. DNA cloning is a technique for reproducing DNA fragments. DNA cloning allows a fragment of DNA or RNA sequence to be selected and reproduced in a required amount. This slide shows the 5 main steps of DNA cloning. Step 1. The gene of interest is cut with a restriction enzyme. Step 2. The host plasmid is cut with the same restriction enzymes. Step 3. Gene is inserted into plasmid and ligated with DNA ligase. Step 4. Transformation of ligation products. And step 5. Growth of transformed colonies on selective media. Steps involved in DNA cloning involves the following steps: restriction enzyme digestion of DNA sample, RE digestion of DNA plasmid vector, ligation of gene of interest and DNA plasmid vector, transformation of the ligated products with the help of DNA ligase, and growth on agar plates with selection of antibiotic resistance. We will look at these steps in detail in the following slides. Step 1. Restriction enzyme digestion of DNA analysis. The gene of interest is cut with the help of restriction enzyme. A restriction enzyme is a DNA cutting enzyme that recognizes a specific target sequence and cuts DNA into 2 pieces at or near that site. Restriction enzymes produces sequence with either sticky ends or blunt ends. Restriction enzymes are present in bacteria as a part of their defense mechanism against phages. We come to the next step, step 2, which is the restriction enzyme digestion of plasmid DNA. The plasmid DNA, as shown here, is cut with the same restriction enzyme which has been used to cut the DNA sample as discussed in the previous slide. This is done so that the sticky ends or blunt ends are able to bind to each other. Step 3, which is the ligation of the DNA sample and plasmid DNA. Ligation is the process of joining 2 pieces of DNA through the formation of a covalent bond. DNA ligases is the enzyme that is used to catalyze this reaction. It requires ATP. In the figure that we see here, the gene of interest which has been removed from a particular DNA is inserted into this plasmid through a DNA ligase and the presence of ATP, resulting in the formation of a recombinant DNA. Step 4. Transformation of ligated products. The process of uptake of foreign DNA into cells is called transformation. There are 2 methods for transforming bacteria. The first is a chemical method utilizing calcium chloride and heat shock to promote DNA entry into cells. A second method is called electroporation based on the electric charge to facilitate DNA uptake. Students, we have now reached midway into this lecture. So far, we have looked at what is DNA cloning and what are the various steps involved in DNA cloning. We looked at the steps in detail. We looked at how restriction enzyme digestion of DNA occurs, the restriction enzyme digestion of the DNA plasmid vector takes place, how these two products are ligated with the help of ligase, the transformation of the ligation products with the help of DNA ligase. Let us now see the various types of transformation, the chemical transformation and electroporation. Chemical transformation. This is one of the ways of transforming the ligated DNA into cells of interest. The log phase E. coli culture is centrifuged, resuspended into calcium chloride solution and chilled on ice. The chilled components are then aliquoted into Eppendorfs and stored at -80°C. Plasmid DNA is added into each Eppendorf, chilled on ice, followed by providing them a heat shock at 42°C in a water bath. It is then plated on agar plates having ampicillin. Electroporation is another way of transforming the ligated DNA products. Log phase E. coli culture is centrifuged and the pellet is resuspended in sterile water. After repeated chilling and resuspension in sterile water, plasmid DNA is added immediately, followed by the provision of an electric shock, and saline buffer is added and plated on agar plates containing ampicillin. Step 5. Growth of transformed colonies on selective N-agar plates. The transformed colonies can be isolated using selective media with antibiotic added to the medium. The non-transformed host cells will grow on the plates containing the antibiotic with blue color, whereas transformed host cells will grow on antibiotic containing plates with white color. These colonies can be picked up and the DNA fragment of interest can be reproduced then in large number. This slide gives a summary of the gene cloning that we have discussed so far. The target gene, for example an insulin gene, is digested with restriction enzyme. A plasmid is also digested with restriction enzyme, and both these components are joined with a DNA ligase to form the recombinant plasmid. The recombinant plasmid is then transformed into another cell which takes up this plasmid and which can multiply containing several copies of these genes. Overview of DNA cloning. In a typical cloning experiment, a target gene is inserted in a plasmid DNA. The plasmid DNA is introduced into bacteria via a process called transformation, and bacteria carrying the plasmid are selected using antibiotics. Bacteria with the correct plasmid are used to make more plasmid DNA, or in some cases induced to express the gene and make protein. Types of cloning vectors. There are different types of cloning vectors used to carry the gene of interest. The cloning vector can be selected based on the size of the fragment DNA to be inserted in the host cell. Different types of cloning vectors are shown in the table below. These include plasmid, which can accept an insert of size 15 kb, a bacteriophage that can accept an insert of size 90 kb, bacterial artificial chromosome that can accept inserts of size 100 to 500 kb, and yeast artificial chromosome that can accept inserts of size in the range of 250 to 2000 kb. Applications. DNA cloning is the first step in the experiments where we require the following applications: production of DNA libraries, polymerase chain reaction, DNA sequencing. Amplification of DNA sequences in large numbers. A single DNA molecule can be amplified and can further be applied for sequencing studies, manipulating mutagenesis or engineering, and expressing for the generation of proteins. In the last section, we saw different types of transformation processes, types of cloning vectors, and applications of DNA cloning. We have now reached the end of this lecture. Let us do a short summary of what we have learnt today in this lecture. DNA cloning is a technique of reproducing DNA fragments. Cloning means cutting a piece of DNA from one organism and inserting it into a cloning vector where it can be replicated by another host organism. DNA cloning involves the following steps: restriction enzyme digestion of DNA sample to pick out the gene of interest, restriction enzyme digestion of DNA plasmid vector, ligation of the gene of interest and DNA plasmid vector using enzyme ligase, transformation with the ligation products with the help of DNA ligase, growth on agar plates with selection for antibiotic resistance. Types of cloning vectors are plasmids, bacteriophages, artificial chromosomes. DNA cloning technique is applied in the production of DNA libraries, polymerase chain reactions, DNA sequencing, amplification of DNA sequences, overexpression of a gene and genetic engineering. Thank you for watching this lecture on DNA cloning.